Oh, and I want to introduce Wan Ling, Zhang, who's here, and I don't think she's been here before. Wan Ling Zhang is a also a human researcher like me. She's a developmental psychologist, and she's been very involved with Zhang and me in the work that we're going to be talking about. Especially, she's really taking the lead on moving the back translation into humans with Zung. So just wanted to introduce her.
Thank
Um,
you. Thank you, Ellen. And it's nice being
Hi.
here to learn from all of you guys about the work that you're doing. Thanks.
So what we're doing today is I'm going to give about a 20-minute talk, and then Zung's going to give a 20-minute talk. We were hoping to leave a lot of time for questions and discussion. This is a very much a mouthful of a title, using frustration studies to study, too many studies, the pathophysiology of pediatric irritability and facilitate development of novel treatments. But I think it will become clear what it is I'm going to talk about in the next few slides. So just bear with me if you would. I don't have any conflicts to disclose. All my work is funded by the intramural program. Oh, sorry about that, of the NIMH. So these are sort of the guiding principles of this talk, and a lot of it won't be news to you, but just sorry, to kind of be sure we're talking about the right similar things. Understanding the brain mechanisms of irritability, and I'm going to be defining irritability for you and giving you a lot of information about what it is. But understanding the brain mechanisms will enable the identification of therapeutic targets. And as all of you know, cross-species work is needed to close the gap between non-invasive macroscopic descriptions of the brain, in other words, human neuroimaging, and invasive microscopic descriptions in animal models. Now, I'm going to show you data. It's actually, some of it's Wan Ling's data, demonstrating that youth with irritability have aberrant responses to frustration. And I don't have to tell this group that frustration can be defined as emotional and behavioral response to emission of an expected reward. And I also don't need to remind you folks that animals experience frustration. So you have a situation here where this deficit in responses to this very common circumstance, emotional context, if you will, of frustration, is cross-species. And that gives you the opportunity to do cross-species studies of frustration. And what basically we're working with is the idea that these cross-species studies can contribute significantly to our understanding of the neurobiology of irritability, and thereby hasten the identification of therapeutic targets. I want to emphasize that this is a relatively rare and important opportunity that we have. Irritability is very common and impairing, as I'll show you in just a moment. There are no firmly established evidence-based treatment. This is an area where you've got like a huge clinical problem and very little is known about treatment. And as I was saying, you've got this cross-species emotional behavioral response of frustration, and those responses are central to the pathophysiology of this very common clinical condition. This isn't unique. You have a similar situation in substance abuse and anxiety, but for example, in psychosis or major depression, you don't. So this is not an opportunity which comes along every day. And my pitch is that it makes a lot of sense, therefore, to try to capitalize on it. So here's the outline. I'm going to tell you some basics about clinical irritability, about a model of irritability based on aberrant responses to frustration that we proposed several years ago, and talk then about clinical studies of frustration and irritability. Of course, then Zung will pick it up with the rodent work. So there are a number of different definitions of irritability, but we tend to define it as increased proneness to anger relative to peers, that may reach a pathological extent. So this isn't what we call an individual differences definition. I'll have more to say about that. But so irritability, people can have, there's a whole dimension where you can line everybody up in terms of their proneness to anger. And these are folks at one end of the spectrum, basically. It's one of the most common reasons that children present for assessment and treatment, particularly in emergency rooms. It manifests in two sorts of ways. Developmentally inappropriate temper outbursts. This is really kind of the hallmark symptom of it. This is what parents and children, it's most salient for them. This is what treatments really especially need to address. There's also tends to go along with sort of a chronically angry, grumpy kind of demeanor. It's present cross-culturally. It's present across a number of different diagnoses. It's particularly common in ADHD, where perhaps up to somewhere between 30 to 50% of youth with ADHD have clinically impairing irritability. And as I said, it's very impairing. For the family, it's got a lot of consequences. They can't invite people over. For the child, they don't have normal peer relationships. It's moderately heritable. Okay, so this is a level of heritability that's roughly equivalent to what you see in anxiety disorders and what you see in major depression. It's less heritable than, say, schizophrenia, autism, ADHD, but it definitely is heritable. Furthermore, it has, oops, sorry, I somehow can't, I'm having a little bit of trouble here. Let's go back. Okay. It predicts, irritability in youth predicts anxiety, unipolar depression, suicidality, and decreased educational and economic achievement in adulthood. So it not only has immediate consequences in terms of function, but it also has some very important long-term consequences. Just very briefly about treatment, there are no firmly established evidence-based treatments. There is an FDA indication for aripiprazole and risperidone, which are some what are called second-generation atypical antipsychotics for irritability, but only in the setting of autism, okay, not in more general settings. And a network analysis of 55 studies showed that this class of medication is effective in reducing so-called disruptive behavior, which includes irritability. But there's a lot of concern about long and short-term side effects of these medications, particularly in children. Metabolic side effects, they gain a lot of weight, tardive dyskinesia, which is a movement disorder which may not be reversible. So there are several other possible medication alternatives. Stimulants in use with ADHD with irritability. The stimulants that are used to treat the ADHD seem to also possibly have a good therapeutic effect on the irritability, although that's only been demonstrated in post hoc analyses. Serotonergic reuptake inhibitors, we did a study, citalopram, and methylphenidate in youth with ADHD and irritability. Oxytocin, I sent this paper around to you folks. And psychotherapies are very important. Parent management training being the most, has the most data behind it, where you teach parents to kind of focus on the positive, not so much the negative, use reward rather than punishment, be consistent, things like that. Okay, so that's just sort of some basics as to the clinical phenomenon that we're dealing with. So as I'll show you, irritability typically manifests in the context of frustration. And so back in, I guess, 2017, we took Amsel's model, and this is a little graphic of Amsel's model, which I don't have to explain to you. Then we adapted it. And we said there are some, and of course, we all know that Amsel's model, this is normative behavior, okay? Here, we're talking about pathophysiology, Okay, we're talking about psychopathology. So we're saying there's kind of the magic here of these neural and behavioral alterations such that when a youth experiences blocked goal attainment, like losing at a computer game, then the frustration that they experience relative to a non-irritable youth is increased. Their anger is increased, their frustration is increased, and they therefore have exaggerated increase in motor activity and aggression. And that, in essence, is the temper outburst. That is the hallmark symptom of irritability. And so this is the very simple model that we proposed in 2017. So now for some clinical data, and here, hello, Wan Ling. So Wan Ling
Can
did
I ask a quick question, Ellen? I'm so sorry.
sure,
You go
sure,
back
sure,
to the last slide?
sure. This one, yes.
Mm-hmm. So I'm curious, how come in the normal, so in the normal model, basically frustration occurs independently from frustration, but not during the temper outburst, is that right?
Well, so when you say in the normal model,
Sorry, instrumental learning, maybe?
well, we don't know where the alteration is. It could be a deficit in instrumental learning. It could be what's going on in here. I mean, this is really very sort of schematic. And, we have done some looking at, we have a study now actually that's designed to test whether instrumental learning, whether there are any differences in instrumental learning between, I'm going to talk in categories, but we really use these as dimensional variables, between irritable and non-irritable youth, either at baseline or their differences in instrumental learning or after frustration.
Okay,
So we're actually doing a study of that right now.
okay.
But it's also possible, and actually I'll show you an imaging study that does to a certain extent perhaps address some of this.
Okay.
All
Thank you.
right, about what might be going on here in the temper outburst.
Okay, thank you. I was just curious about how come, like in the instrumental learning segment, the frustration's isolated, whereas in the temper outburst, it seems like anger and frustration kind of go hand in hand.
Oh,
Yeah,
because we're not necessarily in a position to say that rodents are angry.
okay, got it. That's what I was wondering. Is it just because it's animals, or is it?
Yeah, just because it's animals.
Okay. I got it. Thank you.
Yeah, yeah. Okay, so basically, this is ecological momentary assessment study where the children and actually their parents have smartphones, and they receive prompts 3 times a day for 7 days asking, and here's the sample size, and it was enriched for irritability, asking how they were feeling, what their emotion was. And they were given, these are, they could select any of these. Here's frustration, grouchy, mood change, worry, unfair. And it's a network analysis. So the fatter the line, the thicker the line, the greater the correlation between the nodes. And you can see just by looking at it that frustration is a central node in this network because it has the strongest associations with all the other emotions than any of the other emotions. So it's a central node in the symptom network. And it also, interestingly enough, I'm not showing you these data, but the longitudinal data show that frustration at one time point predicts a mood change at the next time point. So frustration seems to be in some ways driving a lot of what's going on in these youth are enriched for irritability, speaking at an emotional level. Now,
Can I ask a question about that?
Sure.
Tell us exactly what the correlation, how the correlation is obtained. So you ask this at time t, and you ask again at time t+1, and then you correlate the two states. Is that what it is?
Well, no, and Wan Ling, you should jump in here. These are all cross-sectional, okay? So at time one, these are all at time one, and you look at the correlation. This is time one and time two. Okay,
Yeah, and
so
there are
for-
21 time points. So if
Yeah,
you
'cause
think
it's
about
three
three
times
times a day for 7
7
days.
days.
Mhm.
Okay, so now we you
Ellie?
know, yes.
I'm
I'm sorry,
ju-
by the way, I can't see you folks, so do just yell out.
Okay, good. So just a quick clarification on the model there. So they expect that they will not have access, or is it something expected or unexpected? So
It's
they-
an unexpected omission. It's an unexpected, and I'll show you exactly how we do that.
and
when I
that's
talk
3
about the
times per day, you said?
No. Those are different no, this is not a reward or anything. This is just asking them how are they feeling as they go about their daily life. Okay?
Okay,
In the model, I'll be showing you how we induce this experimentally in the scanner. Okay? That's
Okay.
where I'm going now. Okay?
good.
Okay.
Sorry,
Um,
and then real quick,
yeah.
really quick, did you guys probe for positive emotions or as well, or was it only these symptomatic negative emotions here?
Wan Ling, we don't have positive in this, right? Or do we?
Yeah, there's a few positive items asking whether they're happy. And I can't remember, there's another one. But
We can, yeah.
yeah, in here, we only model irritability related symptoms.
Got it. But they
So
were
that
asked,
gets,
so they were asked, right? The positive
Mhm, mhm.
emotions, they just weren't necessarily significant
Related,
or whatever.
yep.
Okay, got it.
we can talk more about that. If you're talking about people who go both into, have a high tendency to go both into negative valence states and positive valence states, then you're talking about lability. Okay, irritability, you're talking about the going into negative valence states. Now there is a correlation. I mean, we can talk more about that, but okay, so now if we're going to do cross species work and we're going to study frustration, we can't just be asking our humans how frustrated are you, because then that we're not going to be able to map on to rodent behavior. So what else do we have at our disposal? And there's actigraphy, which is, you know, wrist measurements of activity. Heart rate and heart rate variability, which is a measure of arousal. Skin conductance, which is a measure of arousal. Face expression, anger in particular, and voice. The rate, tone, and volume of voice is often how you can tell if somebody's very frustrated. In a temper outburst, if you think about it, they're usually yelling, right? There's also, we do use in the scanner and the clinic dynamometers, which basically are button presses that have strain gauges. So we can measure how hard the press is. And you can see here, and I'll be showing you a paradigm just like this, where they're learning a reward game and they're getting regularly rewarded. Then this is the amount of force they use. When they're playing a rigged game, hence they're getting frustrated, this is the amount of force they use. So you can see that that is a measure of frustration. So now let me talk about in-scanner frustration tasks. We evoke frustration in the scanner. I mean, this is again what's really interesting and important, that we can evoke this frustration, which is so central to the pathophysiology. We can actually experimentally evoke it in the scanner, and we can study the brain-wide circuit level responses in real time. We're studying individual differences here in the neural and behavioral responses to frustration when we want to understand irritability. Here's the individual difference question. Why do some youth have difficulty tolerating frustration and others don't? Why are some youth irritable and others aren't? And we really do need more animal work on individual differences, not only in frustration, but really throughout the animal literature. There is some, for sure. But what clinicians really care about is individual differences. Now, how do we frustrate? Most of the work thus far has been done with rigged cognitive tasks, but folks like Wan Ling are developing paradigms to evoke frustration in a social context, which is important because most frustration occurs that way. A lot of challenges that we can talk about. So this is the frustration paradigm that I'll be showing you. This is to show you, and this is self-reported frustration, that it really does induce frustration. You can see it here. It's a large effect size and it's reproducible, okay, which is important with this kind of thing. This is the paper that has the data that I'm going to be showing you. It's 66 kids, so it's still really pilot work. You can see their age. They have a number of different diagnoses, but it's all enriched for irritability. And we do the analyses focusing on ratings of irritability that are obtained from the child and the parent over a period of time, but in particular before the scanning. So before they go into the scanner, they do, it's actually an affective Posner task for those of you who know that, but it's very easy. And every time they do a trial, they win money. Okay, so they playing this easy game. It's like they get 95% correct and they win a bunch of money. We put them in the scanner and we do so-called resting state scanning. I don't know if folks know what that is, but basically it's they're just lying in the scanner, and the purpose of it is to look at the intrinsic connectivity between brain regions. Okay, it's sort of like a baseline measure of the brain when you're not doing any active task. We then have them do this same task, but this time it's rigged. And 60% of the time when they get it correct, we tell them you're wrong, you're too slow. 40, and they lose money. 40% of the time they get the reward. Okay, so this is the frustration induction. And then afterwards, we do resting state again. So they're there at rest, not doing any task. So what we did was we looked at every one of the task conditions, and there's 1, 2, 3, 4, 5, 6. But the data, the findings, I'm going to focus here because this is what the findings are. So this is the brain at baseline before frustration, not doing anything active. And this is the brain right after frustration, which we can call recovery. And again, not doing anything. And all of the significant findings were in the contrast between this and this, or all of the significant findings that were related to irritability. Okay, individual differences, again, related to irritability, are in these two conditions. Very quickly, this also is a network analysis. In this instance, the nodes in the network are brain regions, and the edges is the connectivity. That's basically the correlation in the fMRI activity between the two nodes. That's what an edge is here. And the particular network variable that we looked at is called global efficiency. And before I give you the definition, etc., just look at these two. It's these two modules. Modules, the brain, I should have said, the brain network is made up of multiple modules, with each node being a brain region. But you can see the difference between a low efficiency and a high efficiency module, right, in terms of the number of edges and the pattern. Global efficiency is the capacity to exchange information within a module. It's associated with cognitive processing and with adaptation to emotional stimuli, such as frustration. And here we did it in a prediction network. We did a prediction analysis with a train test held out kind of framework. So our question was global efficiency in which brain circuits and which task conditions predict irritability. I told you the task conditions. It's pre-task versus post-task resting state. So these are the same figure of the brain. It's just tipped a little bit differently. And what you're predicting here, this is a child's measure of their own irritability. This is the parent's measure of the child's irritability. Okay. And here, this is a frontal limbic network in pink here, and it's very much a temporal lobe network. Okay, there does have this node here frontally, but it's both cortical and subcortical temporal lobe. So it has temporal cortex, it has amygdala, hippocampus. Of course, so it's, like a limbic, very much a kind of limbic network. And basically what we're seeing here is that in the post-recovery, in the post-frustration, the more irritable you are, the less efficiency you have in the limbic system. Okay. The parent RE, so this is the parent rating of the child's irritability. That is predicted by this network, which is a somatomotor parietal network. And what we could think about is when a child has a temper outburst, there's a lot going on motor-wise. I mean, this is just hypothesizing. So it has something to do with what the parent sees motorically. And this here, I mean, this is subcortical here. It's very much in ventral PFC. It's caudate and putamen. It's very much of a reward network. So we have these three reward networks that are associated with irritability, that function efficiency in these three networks right after frustration is associated with irritability. It is not associated with anxiety, inattention, or hyperactivity. So it's relatively specific. Now it's a small sample, so this could be a type 2 error, but there it is. So the clinical implications of this.
Ellen,
Yes.
one more question here. Why
Sure.
are you comparing left and right hemisphere here instead of having?
We're not.
Oh, because if
We're not.
okay, I was looking at the image and gave me
Yeah,
the
yeah.
impression it was one side and the other.
No. No. That's actually the same hemisphere. It's just so you can see the bottom of it, the more ventral part versus the more dorsal part. That's all. It's the same, it's the exact same side of the brain, actually. It's
Can you go
not.
back to the slide? I'm confused then with
Sure.
the positions then, because yeah, so the top one, I'm seeing the rostral part of the brain facing the right side, right?
Right.
The other
right.
one, I see the opposite. So it's hard for me to make a comparison where the dots have changed from one side to the other. So
I
my
can give you,
understanding,
yeah.
I'm seeing, so the bottom is the post task, right?
No, they're both post-task. I'm
They're
sorry, they're
both
both
post-task. So
post-task.
they're both the comparison of how much connection you have pre to post-task. Is that what you're?
Exactly.
Mm,
Exactly. That's exactly what they are.
okay,
I can give, and I can, if you look at the paper, I mean, there's way more detail about, it'll tell you in the paper, it'll tell you what all the nodes are and, how we derive the networks and which ones change over the course.
Uh-huh.
I'm giving you a very, very
But what's
narrow.
what's the
Uh-huh.
difference between the top and the bottom? Because they seem to be very different in terms of the way the different structures are connected. There are structures, there are
I'm sorry.
nodes that are isolated in the bottom, for example.
Oh, God. Excuse me a second and let me
So
get this.
what's the difference between the
It's
top
just
and
a
the
different, it's not, they're not different. They're just different views of the same data. They're
Well,
not different.
you have right there in the black, you have in the bottom, you have 1.1 node that's isolated from the rest on the other
And
one.
that is, I believe, let's see, it would be, that is, I think this one, they're sort of angled differently.
Okay, well,
Or no, actually, I think it's this one. They're angled differently. It's this one. It's the same data.
it's the same data.
It's the same data.
Okay.
Okay. Okay,
That's the cingulate that you have there in the bottom is a cingulate cortex that you have, anterior cingulate cortex that you see that in the bottom image that you see a lot of activation, but you don't see on top in pink. In the this one, yeah.
oh, no, that's dorsal, frontal.
Okay.
So where-
Dorsal medial prefrontal cortex.
Yes, dorsal medial.
where,
Dorsal medial.
where is that point in the top graph? That point that's so divergent.
That's part of why it's you can't see it. It's behind here.
Okay,
It's behind. That's why. That's part of why they're showing the two. We're showing the different angles because you can't see it there.
Yeah.
In that angle. I just want to be sure to give Zhang her due. I think this might be my last slide. Irritability is associated with neural dysfunction after frustration, suggesting that youth with irritability have difficulty recovering from frustration. And this is another study from a different group using a very different methodology, which found some similar findings. Aberrant information processing after frustration in 3 specific neural circuits, these 3, is associated with irritability. And both of these points suggest potential mechanistic targets for novel interventions when you want to intervene, which is after frustration, and where you might want to intervene. Again, very preliminary pilot, but just an idea about how you can think about this. And that's the collaborators. And there we go. So I think I really want to get to, how do I stop sharing? Here we go. Yes. And I'm happy to take more questions. I just don't want to steal Zung's time.
I would love to hear more details about the task that people are laying down there in the MRI, and then how is the exact protocol for this?
Let me put the reference in the chat because this is published.
Okay.
So I'll put the reference in the chat. But I'm happy to talk about it, at any point, but also you can see a lot of detail, obviously, in the.
I can take a look at the manuscript, no problem.
Yeah.
Okay.
Over to you, Zung.
Okay, all right. Can you hear me? Can you hear me? Yeah? Okay,
Yes.
great. Yeah, I have been having problems with that. Okay, so Alan has set up the stage for me, so I can skip a lot of this background. So basically, yes, we are really interested in irritability, to study new mechanisms of irritability. I'm just trying to put on the okay. And then as Alan said, frustration, irritability is expressed as a decrease threshold, increase of proneness to anger-induced frustration. And then irritability has a severe and long-lasting impact on children. So how do we study?
Sorry, I'm so sorry. The slides haven't changed for me.
Oh, okay. So let me see. Let me stop sharing. I'll stop sharing and start sharing again and see if we can fix the problem. Okay, maybe I stay on this mode. It's not ideal, but now you can see. Can you see the?
Yeah, can you try like presenting and moving to
Yeah.
another tab?
Yeah, present the PowerPoint.
Okay.
Click on that little cap.
Doesn't allow me to do the presentation mode. It doesn't allow me to. Okay. Yes. Yes. Question. Okay, now I can do the slide show, and can you see my slide?
Yes, perfect.
Yes.
Does it change?
Yes.
Okay, great. Okay, now we're back. Okay, so then how do we study irritability in the mice? So as Alan said, irritability, a frustrating non-reward, which is emotional and behavior response to omission or expected reward, is really the key trigger to irritability. And then FNR is a revolutionary concern. So it's present in both humans and mice. So we can use FNR as a cross-species construct to study irritability in mice. And in addition, the behavior expression of irritability in humans is a higher motor activity and aggression. And again, we can use the same behavior measures in mice to analyze their irritability-like behavior. So, and we have talked a lot about Amsel, who is the first person have noted the phenomenon of FNR. So Amsel has studied FNR using this double runway in rats, and then Amsel has noted FNR has an effect on behavior. So FNR is motivational, so invigorate behavior. After the rats don't get expected reward, the rat will run faster than if they get reward. And then the FNR can increase the resistance to extinction of learned behavior. And also Amsel noticed that frustration effect is greater when there is a secondary reinforcing stimuli. So these are the our starting points, the background of our study. So okay. I think the slide is. In order to study FNR in mice, first off, we decided to design an animal behavior paradigm. We don't want to use the double runway or the output conditioning, output chamber as other people have used before. The reason for that is, like Alan said, irritability is really diagnosis made in children. We want to study this in juvenile mice. The juvenile mice is very small. They may not have a strong muscle to do a strenuous task. And secondly, the mice have a really shortened lifetime comparing to humans. So we essentially only have about 3 to 4 weeks to do our experiments. And then if for people familiar with the Amsel and then the open learning, those paradigms often take weeks of training. So we really want to have a very short protocol so we can complete the training and testing. And then also potentially in future, we want to do surgery in the animals for optogenetics, chemogenetics, and circuit mapping experiment. We really want to finish everything within 3 to 4 weeks. So we have a short period time to work with. So then to achieve this goal, we designed a behavior paradigm. We call it alternate poking reward omission, or APRO. So this is a very simple paradigm. So it's done in a rectangular box. At each end of the box, there is a water port. And then the mice, first of the mice are water restricted for 3 days. So they really have motivation to get water in this box. So the mice have to poke these 2 ports alternately to get the water reward. And when they poke the correct port, in addition to water reward, they will also LED light will light on to be used as secondary reinforcing stimulus. So we train the mice for only 3 days. the mice can learn this task very quickly. And then we reduce the reward to 50% initially, then 80%. So, sorry, we reduce to 50% and then the reward is reduced to 20%. So 80% of the time, the mice will not receive reward on day 5, even they have poked the correct port. So this is our schedule. So for the 3 days of training, and then day 4 and day 5 is a testing with a reward omission, and then day 6 is testing for extinction. So within the 3 days, the mice can learn this task very quickly. By day 3, they can do this alternation almost with 100% success rate. And then when we look at this, the behavior of the mice, this is control mice. The mice receive 100% reward. So you can see the mice are running back and forth, going for these 2 ports. When we look at the mice which have been through the FNR paradigm, so the mice are running faster. They are zipping from one side to another side. So they don't get, this is a video taken on day 5. So they only get 20%, only 20% of the time for correct poking, they will receive reward. So they run faster. So the running speed is faster for the FNR group compared to the control group on day 5. And also as a result of the faster running, they also visit the ports more times than in control animals. So this is consistent with Ansell's observation that frustration or reward omission make animal running faster. So then
Just a question. The mice have to go to the right to be able to get water on the left?
The mice have to go to left first and then right, left, right, left, right.
Do they get water on the right?
Yeah, both sides, both left and right has the water.
Okay.
Yeah. If they stay on the same side, they're not going to get the reward. They have to alternate to get reward.
Can you also clarify the cues that I got lost? You said you have 2 reinforcers. Do you have any cues that you know?
The cue is the LED light. You can see when the I think you should be able to see the light is on. When the mice poke the port, there's a little LED turns on.
So they nose poke and then the LED turns on in the other port where they're gonna retrieve the water.
So the LED is on only if they do the correct alternation. So if they poke the same port, the LED is not on and the water is not going to be delivered.
So the LED is a signal for the availability of reward. Is that correct? They know there is reward if the LED is on.
That's on the day 1 to day 3, but on day 5, even the LED is on, they may not get reward because there's 80% of the time the LED is on, but the animal will not get reward. This is why they get frustrated.
Good. Yeah. During the training, the LED means there is reward available.
Yes. Yes.
Good. Thank you.
Yeah. Mm-hmm. Okay. So it seems like the mice are really getting frustrated, and then we want to we use other behavior tests to look at the behavior of the animal. So basically, so we put animal in this April paradigm. After day 5, they receive 80% of reward omission. We run a battery of a behavior task. So we tested for locomotion, aggression, anxiety, depression-like behavior, and sociability. So there are a total of 6 tasks, 6 tests, and then each test, we only use 1 cohort of animal, and the test is done like within 30 minutes after animal have finished the frustration session. So this is a locomotion. So it's so here we have 3 groups. This is a control group. They always receive 100% reward. This is the group which animal only they have been water restricted, but they have not, they also receive 100%, but they have not been, have not experienced any omission. And this is the FNR group. They have experienced omission. And then we look at both male and female. We found that water restriction alone is not sufficient to change anything. But FNR group for both male and female, they have a higher, they have a longer travel distance. So they have a higher locomotion in this open field task. So the center time, the time in the center of the arena is not significant change. We suggest that animal is not, anxiety is not changed in these animals. And then we look at the aggression. So here we use resident intruder task. So basically this is the black mouse is the resident animal, either is a control mouse or the mouse has been FNR. And then we put an intruder. And then you can see the white one is intruder. So usually the resident at this age, the young age, and also C57 black cyst, they are not really aggressive. So they investigate the intruder, and then they they may play with it, do some sniffing, but not really. They're pretty much very peaceful together. But when we look at the FNR mice, they are very aggressive. So they do this attacking a lot, and it's really unusual for the young animal at this age. And then we find, we see the both female and the male mice after FNR paradigm become very aggressive. This is especially unusual for female because female mice, even adult mice, are not aggressive at all. But after the April paradigm, even the female mice become very aggressive. And this is the measurement of the resident intruder task. So there's water restriction alone has no effect on the aggressive behavior. Both male and female after FNR, they decrease, they initiate attack more quickly, and then they attack more, have more attacks, and each attack lasts longer. So all of them showing that the FNR paradigm make the animals more aggressive. And then we, because aggression is a kind of aggressive social interaction, we wonder whether this is FNR has a general effect on sociability. So we do this classical 3-chamber sociability test, and in this test we don't see any difference between control and FNR mice. Both control and FNR mice, they spend more time to investigate the mouse, the chamber which has a mouse, comparing to the chamber which only has the object. So they are more interested in social subject than with non-social subject. And then we also look at the depression.
Excuse me, Zhang, just to clarify, then these animals are they are familiarized with these other social conspecific or not? Is the first time they're seeing them, or they are co-housed with them?
It's a novel mouse. They have not seen each other before.
Okay.
Okay, so then we look at the anxiety and depression-like behavior. So use the elevated zero maze and LiDAR box for anxiety, for swim test, and the circle preference for depression-like behavior. So overall, we don't see any difference in these measurements. So it looks like anxiety, depression have not been changed in these mice. So this is the summary of the behavior. We see that after FNR, the animal become more hyperactive. They have a higher locomotion. They are more aggressive, but the anxiety, depression, and the general sociability have not been changed. So now it seems like our behavior kind of replicate what EMSO and other people have found in the frustrated animals. And it looks like our April paradigm has a selective effect on motor activity and aggression. Then we, the next step is
before you go to, I need to know how you did this. So all these 6 tests that you are showing here, they were administered
Yeah.
immediately after the task that you have?
So it's
And-
like, it's between like 10 to 30 minutes. And then each test using a different cohort. So one animal will only do one test.
Okay, that
Mhm.
was my question. Okay, perfect.
Yeah.
Thank you.
And how long after the frustration do you expose them to each one of these models? And how many omitted trials do you have?
How many omitted trials? So it's about in the day 5, usually they do like around 50 to 80 trials each mice. And then they between the 50 to 80% is omission. So that'll be 40 to 60 omission trials.
Do you think they may get somehow habituated to that? Because if you have your keyboard is not working anymore, initially you get frustrated, you spunk your keyboard because you know the ladder is not working, but then after 1 hour typing and you
So
stop doing that, right? Do you think they get somehow familiarized with the frustration?
But it's
And-
partial reinforcement, so
They,
you don't get,
so the task takes about 10 to 15 minutes, depends on which experiment. And during this period, we don't see any sign of habituation. Animal keep running very fast.
So they keep
And
running fast, even so that from the beginning to the end of the
Yeah.
omission, they do exactly the same, they have
Yeah,
exactly the
yeah.
same behavior.
Yeah.
partial reinforcement, so it's like your keyboard works and doesn't work and works and doesn't work.
I think that still changes, right? The very first time you get much more frustrated than when you know it may fail, right?
But the animal want to drink, want to have water, so they have been water restricted. They really want to drink water, and then because it's a frustration trial, they haven't drink much, even though they have this omission. They
It's
still
just
want
one
to fight.
session of omission, is 1 day, or you have multiple days of omission?
So we do 2 days, but
2
the day
days.
4 and the day 5, yes.
Okay.
So can you clarify? So the omission trials, are those the ones that are the 50% reward?
Omission, so we have day 4 and day 5. Day 4 is a 50%. Day 5 is a 80% omission.
Okay, so it's like progressive omission, right? Like,
Yeah.
okay,
And also it's a progressive, also it's a different schedule. So day 4 is a fixed schedule. So it's every other correct poking is not going to be rewarded. The day 5 is random. So the 80% omission is around randomly, a random
Gotcha.
schedule. Yeah.
okay. Oh, quick question. Do you guys check if they either like complete fewer alternations, or I'm just thinking about like in operant, we would be able to check like if they go for the inactive lever more often. Is there some sort of equivalent you can check for here?
No, actually, no, inactive, you mean in our case, maybe it's the animal, if they poke one that keep poking the same one,
Right, exactly. Have
Yeah,
you
it
guys
doesn't
looked into
do
that?
much. Initially, may does a little bit, but doesn't do much, that's a lot. It just doesn't have reward this side and quickly go to the other side.
Okay, okay.
Mhm.
Thank you.
Okay. So, and then after this, we really want to know, look into the brain to see what's happening in the brain and what's responsible for this frustration effect. So then we take out the brain, remove the brain from the mice before we do any behavior testing, just after frustration. And then we did CFAS. So we cleared the whole brain and then did the CFAS staining. And this is the data, I think this is the brain, brain regions we have a look at. And then we see some regions have significant difference, but we only have 4 and 5 animals in each group. I think we need to increase our sample size to get more significant regions. But so far, looks like the region significantly changed, activated, including thalamus, some cortical area, like a prelimbic, and prepectoral, and the frontal lobe, and also some hippocampal areas. So this is still ongoing. So and then with this result, we kind of also done a similar analysis as Alan has done for his for her MRI data. We look at the co-activation of brain regions based on CFAS. And then we also look at it, we did this co-activation. This is a co-activation matrix, and we calculated modularity, which is similar to what Alan has done for this global efficiency analysis. So here the modularity, the higher modularity means the brain regions more working in clusters, small clusters. And the lower modularity means the brains working in a more global mode. So more brains working together instead of segmented clusters. And then what we find is comparing the control and then frustrated mice, seems like the modularity is decreased, suggesting that the brain in the frustrated mice is working in a more global processing mode. Okay, and then we also see similarly, like Alan said in the fMRI data, we also see reclustering of brains, brain network. So the left side is the control, and the right side is FNR mice. So and these each group is they clustered into like 7 clusters, and this is before, this is control. And then this is the FNR look like, this reconfiguration of the brain regions that are co-activated.
So sorry, you said left is control group and
Left is
right
control, right is a frust, yeah.
is frustrated group. And why are they connected? I don't understand the slide.
So
Sorry.
so this is the line just indicate the same brain region. So this region is in this cluster here, and also in the control, and then also
So they are the same.
Location of the region in the 2 groups. And then these the box, the gray box indicate the cluster. So this is a one cluster. This is another cluster. So like these cluster, these regions, the top region, they stay in the same cluster, but the other regions, they kind of spread out into other clusters after the first animal.
And what is the meaning for the red sign?
The red is this cluster. The color just indicate just these and the red is more spread out into different clusters. The green is also spread out. They just reconfigured.
Next to the name of certain regions, there's like a red mark. I think that's what.
The red. These are the regions that show increased expression in the CFAS data. Significant increase. These are the regions.
The red one?
Yeah, these yeah, with the red tag here. Yeah.
So how do you interpret this? So what is the meaning of an area changing clusters when you go from control to frustrated? How do you interpret it?
It's hard to interpret now. And even these clusters, I tried to like Alan has for the human give give a name for this cluster, like somatum, sensory or motor or reward or limbic system. Even that in mice is not possible, because one brain region has so many functions in the mice. So we don't know what is the general function of each cluster. But we just know that, maybe this is circuit, the brain regions have different, some circuits are activated, so that activated circuits, the regions in the activated circuits are clustered together.
But you have 2 kinds of connections between control and frustration. Some areas don't change clusters.
Some don't change, yeah. Yes.
So what is the interpretation of that? What does it mean that the area changed from one cluster to another when you go from control to frustration?
Maybe these regions they have a similar functions in this frustration paradigm. So for instance, not really sure. For instance, this is maybe our sensory regions. They stay together. And then there's a frustration network, or is a selective activated, some regions is activated, it's not activated in the control animals, but the frustration network is activated after the frustration paradigm. So they stay together. I mean, I really don't have a clear interpretation from this data.
I would think that if an area doesn't change from one condition to the other, you can eliminate that from your theorizing because it's probably not related to frustration, right? It has the same function whether it's in the control or the frustration, so it's not changing. But if it changes, then it might be implicated in frustration. I think that's the interpretation I would draw.
Yeah, that's one possible way of thinking of it.
For
Yeah,
example, you have the blue on the top, but you also have some green on the bottom, so a bunch of green lines that don't change.
yeah. These guys stay together and these guys stay together.
Right, so those, I
Yeah.
would think they're probably unrelated to frustration because nothing changes there. Whatever they were doing before, whatever they're doing in the control condition, they still keep doing it in the frustrated condition, so they're not specific for frustration, I would think.
Yeah, that's possible. That's a possible way of thinking how it works. But right now, I think without knowing the function of these brain regions, it's really hard for us to make any solid
So what you have
conclusion.
outside are dendrograms? Is that what it is? This,
Dendrogram?
yeah.
This is dendrogram, yeah.
So you did cluster analysis.
Yeah, this is hierarchical clustering analysis. Yeah.
Okay.
Mhm. Okay, I think I have a, yeah, so this is the summary, basically. The imaging result, we find 3 brain regions so far are significantly activated by FNR, and it looks like there is reconfiguration in the brain network after FNR. Okay, so the next step, what we want to do is now we have some candidate brain regions that could be potentially involving FNR. So now we have started to record from some brain regions. We are doing multi-electrode recording in the brain regions, and this is very preliminary. We just started this experiment, and this is a preliminary result. So this is for recording from ACC, and this is recording from NAC. And then we choose these regions because they are involved in emotional processing, and they are also in the reward circuit. This is just local field potential from the animals, and this is power spectrum density. And then what we break these PSD into frequency bands, and we see that almost in all frequency band, we see omission increase PSD more than reward. So this is a log scale on the y-axis. So the real number of PSD is change is much larger. So we see almost at all active ACC and NAC. And this is ongoing. We are still doing. And then, so right now we are trying to still increase the sample size of CFAS. And then we also trying some fibrophotometry, electrophysiology, chemo, and optogenetics to test the functional role of the candidate brain regions. And then we are also, like Alan has mentioned, we want to translate the mouse findings to humans. So the mouse, the human measurements have a similar readout as our activity measurement in the electrophysiology in the mice is the MAG. So we are planning to record, scan the human subjects in the MAG scanner to see if we can find a brain region activity change. Okay, so these are the people who are involved in this work. And so these are the 4 fellows in my group who has been working on this project. Xiao is electrophysiologist, Ajaz is done the behavior, and Maxim also done behavior with Ajaz, and Alahan is doing this human MAG study now. And these our collaborators, Alan, Danny, Wanling, and Lucrezia is helping us with the MAG study. Okay. I'll stop sharing so we can see each other better.
Really nice. I thought that, Ellen, tell me if this is correct, but I think especially your side of the research, is it a prime example of taking irritability as an endophenotype in the RDoc system? Is that what it is?
I'm not sure, and different people define, I'd have to go back honestly and look at the endophenotype. I mean, I guess what that would say, here's the issue, is frustration. It's an interesting issue, actually. Is response to frustration heritable?
Well,
If it is, then it would be an endophenotype for irritability, or could be. But I don't know whether response, I would suspect it might be, which is why irritability is heritable. But Wan Ling, did
Yep.
you, yeah, the VCU twin study, is response to frustration heritable?
That is ongoing work. We have not, that's the work that Lance is working on. So
I
we
Oh,
think-
have
that's the, what
not-
he hasn't, yeah.
I
So
think
we
I
don't know
think
that
you-
yet. We do have a twin study, a
I think it would be
behavioral.
impossible to find a behavior like this, or almost any other behavior that doesn't have a genetic component of some degree.
Yeah.
It would be impossible.
Yeah.
Yeah, but that,
No.
I mean that because you had participants in your sample that came from different, that were classified as ADHD and
Yeah, and if you look at the data via using diagnosis, you don't get the findings. It's the irritability, it's not the diagnosis.
right. So because irritability cuts across all these diagnoses, so in that sense, it's an endophenotype.
Oh, yeah, is again, for frustration to technically be response to frustration to be an endophenotype, you'd have to demonstrate heritability. But I think I agree with you that it likely is heritable.
Of course, everything is.
Yeah, there's some preliminary data suggesting that the say reaction time and their frustration rating in the human work, especially in the frustration runs, are much more heritable than the reaction time or accuracy and other things when kids are not frustrated. So it's specific to frustration.
Wan Ling did a twin study. And with that same frustration task outside the scanner, but that's what she's talking about.
Pretty
I
good.
had just one thought about the presentation. First of all, it was awesome. I loved it. I really did. Because we don't really see much of the clinical side of things. And it was so cool to see that the things that we kind of stumbled upon have like their solid base, right? So I'm like, oh wow, that's awesome. Like with the force things that you showed, that they already use them in the clinic. One thought for Zhang, I was curious to see like if inducing other versions in the APRO, if inducing other, like modifying it or extending it in some way, you could alter the movement of the clusters pre and post. So for instance, something me and Dr. Green have thought about is the fact that there's different types of frustration, right? So sometimes you're just inducing annoyance, sometimes you're inducing like reactive aggression. And I just wonder if you had induced some sort of like reactive aggression, would that change which clusters were activated and stuff like that? It would be like a nice extension to it.
Yeah, that's good idea, yes. So the CFAS experiment, the animals, we remove the brain before we do any aggression task. So it's the animal it's not doing active attacking, have not done attacking, yes.
Yeah, I was just saying like in response to actual non-reward though, not like, yeah, but good. All right.
You know,
Zane, can
problems-
you give us some, sorry, Mauricio,
No,
go ahead.
go ahead. No.
So can you give us some details about all the protocol for the CFOS analysis? Because I can see a huge number of brain areas and we are now in the process of analyzing the brain. So you use standard CFOS immunohistochemistry. How do you process the brains? Do you use an automatic procedure to analyze all the CFOs, or do you do it manually?
Yeah, we do the whole brain clearing. I don't know if you do whole brain clearing or you have sessions. We do the whole brain clearing. We use the shield protocol to clear the brain. And then after clear the brain, we did a CFAS staining, and then we use light sheet microscope to take images from the cleared brain. And then our institute has a imaging core facility. They have make a pipeline for doing the data analysis, and so analyze the CFAS from the whole brain imaging, the light sheet imaging, and then do the segmentation of the brain regions based on Allen Brain Institutes, and then at Brain Atlas, and then do the
But
quantitative analysis.
how do you do the segmentation? Do you do it manually, or do you use a software?
It's a pipeline. It's a software. There's we have a code to do that. It's automatic. It's all automatic.
Okay, I wanted to say simply that we ran an experiment in which we downshifted animals from 32% sucrose to water. And then immediately after, we put them in a social interaction situation. And we have done this as a pre-test before doing the downshift, and then as a post-test after the first downshift session. The animals that were trained in the downshift were the animals that were dominant in the pre-test. And we had a control group that always got water.
Mm-hmm.
Right? So in the post-test, the animals in the control group that were dominant in the pre-test were also dominant in the post-test. So that's the control, right?
Yeah.
They were more aggressive.
Mm-hmm.
But the animals that were dominant in the pre-test and underwent downshift, which is frustration, they had no aggression. And the subdominants in the pair did not become more aggressive. So there was no aggression, very little aggression there in those pairs. So the point that I'm trying to make is that apparently losing a reward not always leads to frustration. And I don't know why you have in one situation a kind of aggressive display, in another something that's more like depression.
Mm-hmm.
I don't know what the factor is that makes the behavior go one way or the other.
The downshift is random.
No,
I mean, by trial.
No. It's 2 phases.
10 sessions of 32% sucrose, and then session 11 is just water. It's unexpected and it's consumatory. And so
And
they
you do
inject
this
the water,
once.
but when you put them in a social context, they lose the aggressive behavior that they had to begin with in the pre-test.
And this is you're done in rats, right?
Yes.
Adult rats.
Yes.
Yeah.
And it looks to us, this we published like 20 years ago. It looked to us like it was similar to what happens in animals that are exposed to inescapable shocks. You put them then in a social situation and they behave in a submissive way. They lose the aggression that they used to have. And so it looked like consumatory downshift produced an animal that was on the depressive side rather than on the aggressive side.
Could be locus of control. Like, yeah.
For that, I don't know about. Yeah, I don't know about rats and locus of control in rats.
Really?
Really, yeah. I would
Okay.
see that.
I mean, there's been things done. They've shown that like when rats have the ability to control whether they can escape.
Oh, I see. No, I was thinking of the human research on locus of control, external versus internal. No, yeah.
Oh, okay.
Yeah.
Gotcha.
It might have I don't know. It might have something to do with that, but in a situation like the one Zheng just gave us, just described, it's the same type of control as in mice. So I don't understand why. Perhaps it's a difference
Well,
between consumatory versus anticipatory behavior.
In Zheng's case, though, it's also escalating, right?
So in our case, like as Alan has mentioned, ours is a reward, omission, reward is they are alternating. But in your case, it's a reward and then all reward, and then no reward or downshift.
One transition from high to low.
Yeah,
Just
Yeah.
once you have many.
yeah. We have many. Yes, yeah.
So maybe that's the difference.
Mhm.
Maybe that's the difference. But it was
And-
always puzzling to me.
Sorry, Sen, did you compare females with males, or did you have any difference in terms of CFOS expression? Because I couldn't see if you were presenting data
Yeah.
for female.
Male and females are different. Although they are both aggressive, we just don't have enough male and female. The N number is too small, but we already see difference. Male has some cortical areas, significant regions. So the data that I show you is all male. The females, we don't really have significant regions. No, a couple, very few. It's a lot less, and it's different regions. There's clear male and female difference in terms of brain activity, CFAS.
Any more questions? If not, then I stop the recording. I will share the link with everybody.
Yes,
Thank you.
And that was great. Thank you so much.
yes, very interesting.
Thank
Yes.
you.
Interesting
Thanks,
stuff.
everyone.
Yeah, thank
Good.
you.
Well, thanks for all your questions and stuff. It's really fun.
Yeah, it is. So next time is Masaki's turn.
Yep.
Okay,
May?
a month from today.
So I'm wondering if we could make this time 1 hour later. I mean, I'm starting from 5 a.m. So if possible.
Yes, for me it's okay because we're in
What?
Spain, go to bed very late. So we can meet
Okay.
at 11 p.m. It's okay for us, I think.
Thank you. If
Send,
that might
please,
help.
Masaki, send me an email asking me about the time so that I can coordinate that.
Okay, maybe just okay. Yeah, I will email you. Okay, to
Remind me
coordinate.
because
Yeah.
I will forget. Remind me.
Yeah, sure.
Perfect. Okay,
So-
I'll stop recording.
Okay.